So welcome friend, it's Abinash here. So today's topic is the DNA repair. So basically the DNA repair system is based on four types: direct repair, excision repair, mismatch repair, and recombination repair. The direct repair includes the two types mainly, which is a pyrimidine dimer repair, and another one is your methyl guanine repair. So what happens? So when the pyrimidine dimers which forms, like the thiamines, in presence of UV light, it's get dimerized in the region of 5.6 carbon atom to becomes thiamine dimer, and to becomes one again normal thiamine, it's need to be enzyme photolase and blue light. The blue light is of wavelength 300 to 500 nanometer. So another example of this is your alkyl transferase enzyme presence of conversion of this methylated guanine at 6th position. So enzyme which is being used here is your O6 methyl guanine DNA transferase. This enzyme remove that methyl group to becomes normal GC bonding, and if there is such methylation is found in guanine, then it will forms bond a pair with thymine instead of cytosine. So we need to remove this, not we, the enzyme we need to remove to prevent the such type of mutations, base pair formation. So enzyme is methyl guanine DNA methyl transferase or alkyl transferase simply. So this is an alkyl transferase. So another thing is your next one is your excision repair system. So in excision repair system, so the excision repair system is involves of two types. One is the base excision repair, and the one is your nucleotide excision repair. So here's the diagram which shows the base excision repair system. So here we need to remove the mismatch base pair, where here you can see that this uracil is being bonded with the guanine instead of cytosine. So you have to remove out this uracil from that region. So for that we need the enzyme N-glycosidic bond to be get clipped out by using enzyme DNA glycosylase. This removes that nitrogenous bases without clipping the phosphodiester bond. Remember, without clipping the phosphodiester bond. This phosphodiester bond without that nitrogenous base has called the AP site. That means apurinic and apyrimidinic site to get clipped by AP endonuclease. This endonuclease clips out the breaks the phosphodiester bonds at this AP site, apurinic or apyrimidinic site. So after removing that region, we need the enzyme DNA polymerase 1 and ligase to repair this DNA segment. This is called the base excision repair system. Next one is your nucleotide excision repair system. Here it can act more substantially damaged areas of the DNA. So it is the either side of the DNA lessons is caught. That means you can see here there is a regions of this portion, as you can see, need completely clipped out. This is a lesson. So for this we have to be, for example, is taken here is nucleotide excision repair of pyrimidine dimer. So for this we need enzyme UVRA, UVRB, and UVRC. This is a combination of ABC exonuclease which acts not simultaneously with association with some other proteins of their own. So for example, first thing here, the UVRAB comes together, and the function of UVRA is to incorporate this B on the DNA segment where they need to be get repair. So that means the UVRA recognizes that site of mismatch region. So once the UVR recognizes, it will be UVRB which get attached to this region. Then the UVRA is replaced by UVRC, and this UVRC forms a UVR-BC complex which clips the damaged DNA strand at the 8th phosphodiester bond on the 5th side of the bind, and this is removed out by using the DNA helicase to becomes once again be repaired by using enzyme DNA polymerase and DNA ligase. So in conclusion, the UVR A B comes, B binds, A recognize, and the UVR C clips out, and this clipped out segment is removed by the helicase 2, and then the DNA polymerase 1 and ligase repair this with new bases. Next one is your next one. This is about the mismatch repair system. In the mismatch repair system, there is before this mismatch repair, we have to know about real thing that the parental DNA strand is methylated at some particular region of adenine of the G8AC sequence, which is not a mutation. So when a new strand, a daughter strand, is being synthesized, it lacks that kind of methylation by which the repair system can be done before this methylation has begun. So the 5' G8AC is the sequence where the adenine get methylated to becomes the parent DNA strand formation, and also some cytosine regions like a sequence of CCAGG where the C is not the CC, that means the second C is that region where the methylation occurs. Similarly, the CCAGG and CCTGG is the two sequence where the second cytosine is being methylated to becomes a parental DNA strand. So this methylation is done by enzyme adenine methylase and cytosine methylase respectively. So how this mismatch repair is being carried out? So first thing, there is a requirement of the two proteins, that is your mut H and mut S. Mut S and mut H is responsible for this. The mut S helps to recognize, whereas this mut H causes the cleaving of this segment of the unmethylated region. So once they get cleaved, it's get removed by the using enzyme helicase 2 and exonuclease 1, and this removed segment need to be once again prepared by using the enzyme DNA polymerase 1 and DNA ligase to join those segments. Now come to the recombination repair system. Well, in this recombination repair system, the DNA with the damaged strand is being need to be replaced with a new fragment, a new segment from nearby homologous segment. So the homologous segment has to be get incorporated into this region. For that, we need to be get remove this damaged DNA. The damaged DNA is get removed out, and the removed out region is being filled with that homologous region of this segment to get incorporated. So the actual mechanism is in the next diagram. Here is this figure. You can see here the nick is being done here, and for this nick formation the replication fork will get collapsed, and to repair this replication fork region we need enzyme REC BCD, REC A. Such type of enzyme is being required. So what is the function of this enzyme? Well, the REC BCD is an enzyme which helps to make a 3-dex overhang elongation DNA segment that has to be get embedded into that correct sequence of the parental DNA strand by using REC A. This REC A causes invasion, which is to get forms a D loop or holiday junctions. These holiday junctions will cause the exchange of that segment to becomes from the homologous daughter strand, newly formed daughter strand, so that the segment which is get unable to form that the collapsing of that replication fork won't happen. So after the formation of holiday junction, the holiday junction need to get clipped out So after clicking out, it will gives a replication for 1 second to be get carried out the DNA replication. The last one is the repair of the double strand DNA by using the non-homologous end joining, that is your NHEJ repair system. Another one is your SDR repair system, that means homologous direct repair system. The non-homologous end joining repair system is very fast and very effective, highly efficient, but it's more prone to error. Whereas in case of homologous recombinant DNA repair system SDR, it is very less efficient, but it has less prone to mutations, or we can say that very not at all the mutation is in there. So the non-homologous engineering repair system is required for emergency, for instant repair. So it's found in G0 and G1 phase of the cell cycle, whereas in the homologous direct repair is required at the S and G2 phase of cell cycle. So the basic mechanism here is that you can see the direct strand of the break has the Q80 and Q70 is a heterodimer which recognizes that breaking point of the double stranded break in the DNA. This recognition is then being cleaved by phosphorylated by the enzyme DNA dependent protein kinase CS, which is phosphorylate this region, which need to be then trimmed both the ends by using a protein Artemis. The Artemis clips or trim this both the strand, this region, and then it gets ligated by using DNA ligase 4 and XRCC4 associated. Whereas in the double-stranded break of the break is repaired in the homologous direct repair system is similar to that of the homologous template DNA strand, where the D loop is formed, holiday junction is formed, and it is the complete exchange of that region of the particular segment of the DNA to becomes repair with the actually parental sequence of DNA without causing any kind of mutation. So this is the complete of DNA repair system. Thank you very much.